Hi everyone, this is a chance to go over some of the topics that will be covered in your midterm exam. I do want to advise you that this is not a comprehensive midterm review. It's kind of the bare bones, so make sure that you do go through your book, go through your notes, and study on your own as well. If you just watch this, it's a good stepping point or starting point, but make sure that you also do your own research. Alright, that being said, let's start with the scientific method. This is one of the first things that we covered in week 1. Things you need to know are the steps. There's 5 steps. Make sure that you understand the order they're in and also what each of them means. And then most importantly, focus on the hypothesis. You will have at least 2 questions about what is a hypothesis, how do you test it, and so forth. So make sure you understand that concept. Looking back at your labs, there are several questions about hypotheses, so that might be a good place to start. Another topic I want to focus on is homeostasis. It's important to know the basic concept of homeostasis because it's kind of the basis of everything that science is based on, or biology at least. We also use the word balance when we talk about homeostasis. And I don't want you to confuse balance with being equal. So things need to have a balance. Cells need to have a balance. But that doesn't always mean the same on the inside as on the outside. Depending on what subject you're talking about specifically, that could be different. A lot of things need to maintain a balance that's not equal. So be able to use some examples and understand what that means. Another good way to go over this again is to look at your lab questions, both the pre-lab and post-lab questions. And then dealing with homeostasis, we do mention a lot about positive feedback or negative feedback and what that means. With positive feedback, remember that you are increasing a stimulus. So if something's going up, it's going to keep going up. One of the most commonly used examples is oxytocin during labor. So until that child is born, the oxytocin levels are going to increase, which triggers more of an increase and more of an increase. And then once the delivery happens, everything can kind of bounce back down to normal. Whereas a negative feedback loop, we often use body temperature as an example here. So if your body is cold and you're in a cold environment, your body's going to take the necessary steps to become warm. So you'll start to shiver. You'll feel like you have goosebumps. Those muscles will contract. And your blood will kind of center towards the core of your body to keep it warmer than it would in another situation. And that's an example of the stimulus and your body doing the opposite. So in positive, the stimulus keeps going up. And in negative, the reaction is going to be the opposite of your stimulus. So make sure you check out a couple pictures and examples of that as well. Alright, we did spend a little time on chemical elements and compounds. Most of this information was in your lecture part so far, so look through the online modules for information on this, and also your textbook. Great examples in your textbook. You will have several questions on the basic details, maybe the makeup or what they're used for, of either carbohydrates, lipids, proteins, or nucleic acids. Those are 4 major groups that you'll need to know the basics about. And when we talk about the molecular makeup of these things, the type of bonds can affect its qualities. So look up, if you haven't already, if you're not familiar with these terms, look up a covalent bond, hydrogen bond, ionic bond, and what is similar about those things and what is different, what makes them unique. Alright, moving on to another topic we covered well in lab is pH. What does it mean? What does it do? The very, very basic part of pH is that acids always want to donate their protons. So they have too many and they want to give them away. Bases are the opposite. They don't have enough protons to be stable, so they want to accept it from someone else. So acids and bases get along well because they can both donate and accept. Understand what the 0 to 14 scale is. I think we're all familiar that 7 is neutral, but make sure you know if it's above 7 or below 7, does that mean acid or does that mean base? Make sure you look that up. Alright, starting with the smallest unit of life, we have the cell. And we've talked about the cell quite a bit, especially when we did the lab that covered the osmosis. So we were measuring what goes into and out of a cell. That's called the phospholipid bilayer. Now, things get through by one of two ways. It's either going to be active transport or passive transport. That's one of the things we measured with the dialysis tubing. With passive transport, there is no energy needed. That means no ATP. With active transport, we are trying to push something through, but we need energy to get it there. And that energy always comes in the form of ATP. ATP stands for adenosine triphosphate. There will be at least 2 questions about ATP and how it's used, what it's used for. Make sure you look that up in your book. And as long as we're talking about the cell, we didn't talk about this much in lab, but in your online modules, we did go over the organelles and what they are. So organelles mean the little parts that make up the cell. Could be mitochondria, a nucleus, Golgi body, and so forth. There's lots more. Make sure you have a basic understanding of each one and what its function is. Most importantly, the mitochondria, being the energy source or powerhouse, and the nucleus, which kind of acts as the control center or the brain of the cell. But make sure you're looking up all of the organelles and have an understanding of each of those. Alright, now, coming up on week 5, we're going to be talking more about cell replication. So if you think of it on a big scale, your body is constantly replacing its own cells. And if it wasn't, that means that you would be dying. You would slowly be dying until your cells no longer replicate themselves anymore. So to be alive and to have an organism have life, it needs to continually regenerate its own cells. There's 2 processes that can do this. One is called mitosis and one is meiosis, or can be pronounced meiosis. Mitosis is creating an exact copy of the same cell. So maybe a brain cell is going to split up and it's going to become 2 of the exact same copies. The opposite is meiosis. So with meiosis, you're going to have one cell that actually splits into halves of itself. And that only happens in 2 cases: male sperm and female eggs, or gametes. Because if you think about it, when you're making a new being, if you had a whole cell and another whole cell combined, you would have too much DNA. But if you had a half cell and a half cell combined, then you're creating one full cell, or one full new human being. So double check the differences between the 2, how much DNA is used in mitosis versus meiosis, and how those daughter cells are created. Make sure you know the difference. When it comes to the actual cycle of making these cells, we have several different ways to do it. We have prophase, metaphase, telophase, anaphase. If you haven't already looked at these, make sure you understand how it works. There's a different cycle for mitosis and meiosis. They each have their own way of going about it. There's great pictures in your textbook about this. Definitely check those out. Alright, one thing that gets split up during these cell cycles is the DNA. And we'll talk a lot more about this in week 5 in your week 5 labs. So if we haven't gotten there yet, that's okay, but we need to get there before you take the exam. When it comes to DNA, we know it's found in the nucleus of the cell. With DNA, there's 4 types of nucleotides: A, T, C, and G. Those stand for adenosine, thymine, cytosine and guanine. In RNA, which is a single strand, we have A, U, C, and G, where the U is replaced with uracil. We haven't done this yet if you're not in week 5, but during your week 5 lab, we'll talk about how to transcribe or translate DNA with its matching strand. So A is always going to match with T, C always matches with G, and you'll be asked to actually write this out. Make sure you practice doing this. It's going to be critical. You will have a question where you have to determine which strand matches another strand. And try not to be too concerned about it if it sounds kind of out of your reach, because we will talk about it in lab and it'll make a little more sense at that point. Alright, this is important. You will have several questions that relate to your critical thinking assignment. So if you remember the critical thinking assignment you had about tomato plants, it asked you to identify the independent variable versus the dependent variable, and then several other questions about the scientific experiment that was presented to you. On your midterm, you will have a question that lays out similar examples. You'll be given a scenario, and you'll have to figure out what's dependent and what is independent. And this is, I'm going to take you back. I know if you've seen me in lab, I've given you this example several times, but it's really important. To figure out which is dependent and which is independent, you need to just fill in the blanks. So create this sentence. The dependent variable is dependent on the independent variable. So if we fill in those blanks with the tomato plant assignment, we found out that the size of the tomato depended on the amount of sunlight. Therefore, the dependent variable was the size of the tomato, and the independent was the amount of sunlight. The example you'll be given on the exam is very, very similar to this, so make sure you have a firm understanding of what those questions were asking. Alright, a couple more things. ATP, we mentioned this a little bit already, but you will need to figure out the answer to these 3 questions. What do you need to create ATP? So there's 2 products that you need. What is a byproduct of ATP production? So when you're actually creating it, what is something that's created but kind of left out? So if you're combining 2 products and you actually end up with two end products, what was that other one that we're not using? And then thirdly, where is ATP produced? So where inside the cell, or where inside the human body do we make this stuff? And lastly, we talked about this in Module 4, but the types of tissues. I have several questions on this. Things you need to know are the basic concepts. So what does epithelial tissue do? Is it for support? Is it for protection? Does it provide movement? Think about that for each of these 4 types, and then also understand, is there a lot of matrix or no matrix? What type of cell shape is it? Is it very flat? Is it round? Is it big? And what is the function? This is exactly what we talked about in Module 4 in the Module 4 lab, so make sure that you're going over those answers while you're studying for this exam. Alright, so that kind of wraps up the main points. If you have any questions or something wasn't clear enough in this review, please let me know. I'd like to clear that up before you take the exam. And as always, I'm reachable by phone, email, or however you want to get a hold of me. It's fine with me. I'd love to answer your questions before anything becomes a problem. So I hope this helps, and good luck studying.